Hello and welcome. Today let's talk about the urinary system and we'll check out excretion. Before we jump in here, we can appreciate some pictures in relation to the kidney. Here we can see on the left we've got a normal kidney, and then here on the right we can appreciate a kidney with multiple cysts. So this kidney we see here is a polycystic kidney. And all of these cysts we can appreciate here on the kidneys can lead to the kidneys not functioning properly. Next in here, let's check out the urinary system. I'd like you to know first about the urinary system's functions. So we'll go through and we'll talk about the functions. Here, when we talk about the urinary system, you'll see it's involved in excretion. It is involved in excretion, which is the removal of metabolic waste from the body. Now, this should not be confused with defecation. Because when we talk defecation, defecation is the elimination of feces from the body, which you'll see is a function of the digestive system. Also, I would like you to know when we talk about the undigested food and the bacteria that's going to make up our feces, you have to remember they were never a part of the functioning of the body, right? We ingested them through the oral cavity, they pass through this gastrointestinal tract, and then you can see they're removed. Now, here, when we talk metabolic waste, these are substances that are actually going to be removed from the bloodstream. Now, when we talk about the functions of the urinary system, I would like you to know that the urinary system is going to, number one, produce urine and then conduct it to the outside of the body. Now, we'll see as the urinary system produces urine, it's also going to carry out other functions that are going to help maintain homeostasis. So first here we can see is the excretion of metabolic waste. The excretion of metabolic waste. The kidneys, we'll see, are going to excrete metabolic waste. And most of this metabolic waste is going to be nitrogenous. So first here we can see we have urea. That's going to be the primary nitrogenous waste we're going to see. It's a byproduct of amino acid metabolism. It's going to be made from ammonia and it's released from our amino acids. Second, we can appreciate here we have ammonia. Third, then we have creatinine. Creatinine is going to be the breakdown product of creatine phosphate. And then last we can appreciate here is uric acid. And uric acid is going to be made from nucleotide breakdown. So again, urea is going to be a byproduct of amino acid metabolism. When amino acids get broken down in the liver, ammonia is going to get released. This ammonia is going to be acted on by the liver. The liver is going to quickly combine it with carbon dioxide to produce urea. Because we'll see ammonia is going to be very toxic. And then when we talk about the breakdown of nucleotides, such as those that contain adenine and thymine uric acid, too much uric acid, we'll see, is going to lead to the formation of crystals in our joints. And when we have the formation of crystals in our joints, this is going to lead to a very painful crisis, which is called gout. Gout. The second function that we can check out is going to be osmoregulation. Now, when we talk osmoregulation is going to be the maintenance of a balance of water and salt in our blood. Now, when we talk osmoregulation, through osmoregulation, you'll see the kidneys are going to be involved in regulating blood pressure. And last here we can see the kidneys are also going to help maintain appropriate levels of other ions in the blood too, such as potassium, bicarbonate, and calcium, to mention a few. Third function then we can check out is going to be the regulation of acid-base balance, the maintenance of our acid-base balance, along with the respiratory system. Okay, along with the respiratory system, the kidneys are going to monitor and help keep our blood pH at 7.4. And they will do this by excreting hydrogen ions and by reabsorbing bicarbonate ions. And then we can check out the fourth function. The fourth function of the urinary system is going to be the secretion of hormones, a very important function as well. The kidneys, they release renin, we can see here. Renin is going to be an enzyme that leads to the secretion of the hormone aldosterone. It leads to the secretion of aldosterone from the adrenal cortex. And aldosterone, we have seen before, aldosterone increases the reabsorption of sodium ions by the kidneys we have mentioned. And then the second hormone we can see here is erythropoietin. It's abbreviated as EPO. Erythropoietin stimulates red blood cell production in the presence of reduced oxygen carrying capacity or if our oxygen demands increase. The kidneys, we can see, are also going to be involved in vitamin D activation. They're also involved in helping to activate vitamin D, which then you can see is going to promote calcium absorption, which then promotes calcium absorption. Next, then, let's check out the organs that are going to be involved in helping to make up this urinary system. All right, next, then, when we talk about the urinary system, we'll see the urinary system is going to be made up of, number one, the kidneys. Number two, then we have the ureters. And then number three, we've got the urinary bladder. And then fourth, we'll have the urethra. And we'll look at each of these organs there then in greater detail. And let's start off by talking about the kidneys first. Now, when we talk about the kidneys, I'd like you to know the kidneys are going to be found as a pair. So they are paired organs. They're going to be bean-shaped. They are reddish-brown organs that are going to be found located in the lumbar region. They're going to be found located in the lumbar region of the vertebral column. The kidneys, they're going to be about the size of our fist. They're about the size of our fists. They're going to be covered by peritoneum. So they're found located behind the peritoneum. Since they're found behind the peritoneum, they are considered retroperitoneal organs, along with a whole bunch of other organs. Another famous organ that you could remember is going to be the pancreas. So the pancreas and the kidneys are found behind the peritoneum. We'll see the kidneys are also going to receive protection from the lower rib cage. They will receive protection from the lower rib cage. I'd like you to know that each kidney is going to be covered by a tough, fibrous connective tissue layer that's going to be called the renal capsule. Each kidney is going to be covered by a renal capsule. We'll also see each kidney is going to have a depression. It's going to have what we call a hilum on its concave or medial side. And this hilum is going to be marking where the renal artery enters the kidneys. And it's going to mark where the renal vein and the ureter exit the kidneys. And where the vein and the ureter exit. Next, then, let's check out our ureters. Our ureters. Now, when we talk about the ureters, we'll see the ureters, they are going to function to conduct urine from our kidneys to our urinary bladder. So they conduct urine from the kidneys to the bladder. Our ureters are going to be muscular tubes, and they're about 9 to 10 inches in length. They're about 9 to 10 inches in length and about 5 millimeters in diameter. Each ureter is going to travel down behind the peritoneum, so they are also retroperitoneal organs. They'll travel down from the renal hilum of the kidneys to enter the urinary bladder on its dorsal inferior surface. Now, when we talk about the wall of the ureters, you'll see the wall of each ureter is going to be made up of 3 layers. We will have an inner mucosa, a smooth muscle layer, and an outer fibrous layer of connective tissue. Peristaltic contractions, like we saw in the digestive tract, are going to be responsible for moving urine down these ureters and into the bladder. So this urine is conveyed down the ureters into the bladder by peristaltic waves, by peristalsis. A healthy person, you're going to see, is going to produce anywhere from 1 to 2 liters of urine a day. They will produce 1 to 2 liters of urine a day. Next, then, let's talk about the urinary bladder. Now, when we talk about the urinary bladder, we'll see the urinary bladder is going to function to store urine until this urine can be expelled out of the body. Our urinary bladder is going to be found located in our pelvic cavity. It's going to be found located behind our pubic symphysis. The urinary bladder is going to have 3 openings, or we can say 3 orifices to it, and 2 of these are going to be for the ureters, 1 for each ureter, the right and the left ureter, and 1 opening is going to be for the urethra, for our 1 urethra. And when we talk about our urethra, we'll see it's going to be responsible for draining urine. It will drain urine from this bladder. Next, I'd like you to know the walls of the urinary bladder are going to be expandable, and they are made up of 2 longitudinal muscle layers and a single circular layer. The mucosa is going to be made up of transitional epithelium. It will be transitional epithelium making up the mucosa. And here last we can see we're going to have 2 sphincters that I want you to know about. These sphincters or sphincter muscles are going to be involved in helping to control the release of urine into the urethra. One will see the internal sphincter. It's going to be found around the opening to the urethra. The second, a little bit further down, is called the external sphincter, and it's going to be composed of skeletal muscle, and it's going to be under voluntary control. And it will be under voluntary control. All right, next then let's check out the urethra. Now when we talk about the urethra is going to be a small tube that's going to lead from the urinary bladder to an external opening. It functions to remove urine from the body. It serves to remove urine from the body. Now in females, the urethra is going to be short compared to the male. In the male, the urethra is going to be much longer. We'll see in the female, the reproductive and urinary systems are not connected. They're not connected. They're going to be completely separate. In the male, we'll see the urethra is going to carry urine during urination and semen during ejaculation. And we'll check that out in the reproductive chapter in greater detail. Last, I'd like you to know that the male urethra is going to be encircled by our prostate gland. It's going to be encircled by our prostate gland as the urethra exits the bladder. As the urethra exits the bladder. So we'll have the small miniature donut-shaped gland encircling that part of the urethra. All right, next then let's check all these organs out. So here we can appreciate the right kidney, and we have the left kidney. You can see the right kidney is slightly lower. It's going to be slightly lower because we have our liver that's going to be occupying the space above that right kidney. So it causes this right kidney to be slightly lower compared to the left. And you can see above those kidneys we have the adrenal glands, the suprarenal glands. So next then from the kidneys, this urine is going to travel into the ureters. So here's one ureter. And then here we can see the other ureter. Via peristaltic waves, they're going to take urine down into the urinary bladder. The urinary bladder is going to be a storage tank, and it can then release urine, you can see, through the urethra. So the urethra is going to pass urine to the outside. And then last, we can appreciate here the renal artery and the renal vein on both sides. Next, then let's talk about urination. When we talk urination, I'd like you to know urination is going to be triggered when the urinary bladder is going to fill to about 250 milliliters. What happens is you've got stretch receptors that are going to be found within the bladder. These stretch receptors are going to start sending signals to the spinal cord. And then we have motor nerve impulses that are going to cause the sphincters to relax and our bladder to contract so that urination is going to be possible. Now, in older children and adults, the brain is going to control this reflex. Urination or micturition, we can refer to it as scientifically, again, is going to occur when the bladder fills to about 250 milliliters. So when that bladder fills to about 250 milliliters, here you're going to see we've got all these stretch receptors that are going to be found in the walls. Those stretch receptors are going to start sending signals then to the spinal cord. And then we can see motor nerve impulses coming back from the CNS are going to cause the sphincters to relax and the bladder to contract, allowing us to then expel out the urine. All right, next here then let's talk about the anatomy of the kidneys, and we'll look at excretion then in greater detail here as well. Now, when we talk about the anatomy of the kidneys, here we can see this figure shows us a nice view of the inside of the kidney. So here we can appreciate a whole bunch of different structures we're going to go through and we're going to talk about. First, we can appreciate here we've got the renal artery. Now, this renal artery is going to be responsible for bringing blood into the kidneys. So here you can see it brings blood into the kidneys. It brings blood in all the way to the end. And then here is where we're going to have our filters, our nephrons. You can see that's where they're going to be found. So here you can see in greater detail, they're going to be found connected to the artery and the vein. The vein then is going to be responsible for taking blood back out. So blood is going to be making its way through all these little veins and then eventually into the renal vein and back into our inferior vena cava. And back into our inferior vena cava. And then here we're going to see our waste. Our waste is going to be making its way down then through, or we can say our urine, right? Through this ureter, right through this ureter. So inside of this kidney, we can appreciate we've got our renal pyramids. And we're going to see in between the pyramids, we have our renal columns. The kidneys, we can see, are going to be divided up into three regions. You've got first, outside of here, the cortex. In the renal cortex is where we can appreciate the nephrons. This is where the nephrons are going to be found. And then we've got the renal pyramids here. The renal pyramids are found in the second region. This is the renal medulla. This is all the renal medulla. And then the third region we can appreciate inside of here is the renal pelvis. The renal pelvis. The renal pelvis, you can see, is going to be continuous with the ureter. It's continuous with the ureter. All right, next then let's talk about these nephrons. They're the VIPs here. Let's talk about these nephrons then in greater detail. Each kidney, I'd like you to know, is going to be composed of millions of these tiny blood processing units that we call nephrons. Very important you go to anatomy knowing about nephrons, at least this basic structure. Now, when we talk about nephrons, we'll see each nephron is going to have its own blood supply, and it's going to have two capillary regions, and it will have two capillary regions that we're going to go through and we're going to talk about. So here we can see each of those nephrons. Once they're done processing the blood, whatever they remove out is going to be urine. That will be urine. So you can see right inside of here, urine is going to make its way through and it gets dropped off right inside of that pelvis, right inside the pelvis. So here are those pyramids you see right in here. So you can see these collecting ducts. They're opening right into the pelvis. They open up into the pelvis. And here from the pelvis, then the urine is going to make its way into the ureters as we saw. Same thing we can see here, a nice frontal section of the kidneys. I'd like you to know here, nephrons are going to have their own blood supply, and they have two capillary regions. From now the renal artery, okay, from the renal artery you can see there's going to be an afferent arteriole that's going to lead us to what we call the glomerulus. So right here you can see this tuft of capillaries. This bunch of capillaries is called the glomerulus. The afferent arteriole is going to lead us to the glomerulus. The glomerulus is a knot of capillaries that are going to be found inside this glomerular capsule. That are going to be found inside the glomerular capsule. Now you can see blood leaving the glomerulus. Blood leaving the glomerulus is going to enter into the efferent arteriole. Blood exiting that glomerulus is going to exit via the efferent. So you can think of E for efferent. A for approaching. Approaching what? The glomerulus. Just one way to remember. This efferent arteriole, it's going to guide blood to, we can see here, the peritubular, right? Peritubular, the prefix peri means around or surrounding, tubular capillaries. So the efferent arteriole will lead us to the peritubular capillaries, which are found surrounding, you can see the nephron. From there, blood is going to move into a venule. It will eventually move into a venule. And this venule will join the renal vein, which will empty, we said, into the inferior vena cava. So whatever gets removed is going to make its way into this tubule. Whatever doesn't get removed is going to be sent back in the blood through the efferent arteriole into the peritubular capillaries, and then into a venule and back into the renal vein and the inferior vena cava. So let's go through the different parts of this nephron. The nephron, you can see, is made up of a whole bunch of different parts. First, you're going to have the glomerular capsule. You can see here the glomerular capsule, or we can call it Bowman's capsule. The inner layer of this Bowman's capsule, the inner layer, I want you to know, is going to be made up of these cells that are called podocytes. Podocytes. These podocytes, they are going to cling to these capillary walls. They're going to cling to this glomerular capillaries walls, and they leave these tiny pores. Those pores are going to allow for the passage of small molecules from that glomerulus. Okay, they're going to allow the passage of small molecules from that glomerulus to the inside of this glomerular capsule. So now from here, whatever got filtered out is going to be making its way into the next segment. The next segment that's connected to this glomerular capsule is going to be the PCT, this proximal convoluted tubule. The proximal convoluted tubule then will receive that filtrate. Here in this proximal convoluted tubule, we have cuboidal cells. We're going to have cuboidal epithelial cells that are going to be lining this part of the nephron, and these cells are going to have numerous microvilli. And what these microvilli do is these microvilli are going to increase the surface area, as we have seen before. They're going to increase the surface area for reabsorption. Well, you might ask, wait, why for reabsorption? We just excreted things. Well, in case something made its way out that we didn't want, it can get picked right back up. Also, what happens here, you'll see 65% of the components that get filtered out eventually get picked right back up by this PCT. They get picked right back up by the PCT. Next then, we can see the tube is going to narrow. It's going to narrow and it's going to make a U-turn. And it makes a U-turn, you can see here. And this next segment we've got here is going to be called the loop of Henle. It's called the nephron loop, the loop of the nephron, or the loop of Henle. This loop of Henle has a descending limb and it has an ascending limb. This descending limb, you can see, is all thin. The ascending limb is made up of two segments, a thin segment and a thick segment. It's made up by a thin segment and a thick segment. This loop of Henle is going to be lined by simple squamous epithelium. It'll be lined by simple squamous epithelium. The next segment we can appreciate here after the loop is going to be called the DCT, or the distal convoluted tubule. This DCT, or distal convoluted tubule, it's going to be composed of cuboidal epithelial cells. This DCT, it is not involved in reabsorption. Rather, it helps to move molecules from the blood into the tubule through this process that's called secretion. Secretion. So anything that didn't make its way out here and it's still in the blood, well, we have another chance to take it out. And that chance is here. We can secrete it out into the DCT. If not, then it'll remain inside the bloodstream. The DCTs, the distal convoluted tubules of these nephrons, you can see then are going to make their way into the collecting duct. And here we can see one collecting duct is going to be able to receive many DCTs. One here can receive, another one here, and then we can see we have another one here. So you can say that distal convoluted tubules of several nephrons are going to enter one collecting duct. And then many collecting ducts are going to carry urine to the renal pelvis. They'll carry urine to the renal pelvis. So here you can see that glomerular capsule, and then right on top of here, this is where all those podocytes are going to be found, right on top of that glomerulus. And then next we can see everything here together. So these are all the different segments of the tubules. You can see here's a collecting duct. These are capillaries here. Here we have an ascending limb of a loop of Henle, and we have a descending limb. Remember, we have millions of these tiny blood processing units in each kidney. So there's so many of these different tubules that can appear in one slide. And the last here, I'd like you to be able to appreciate the glomerular capsule and the convoluted tubules. The glomerular capsule and the convoluted tubules always are going to be found lying within that renal cortex. They're always going to be found lying within the renal cortex. Now the loop of Henle, the loop, you can see it dips down into the medulla. It dips down into the medulla. Some loops are going to extend further down into the medulla than others. Now, the collecting ducts we can see, they're mostly located in the medulla, and they're going to help give the renal pyramids their lined appearance. So if we move back in here, you can see right in here, they're going to help give that pyramid this lined appearance that you see right in here. Let's move back even further. We move back even further. Right in here, you can see that lined appearance. That's all thanks to those collecting ducts. And they open up, you can see, right into the pelvis. Right into the pelvis. And from the pelvis, we said urine's going to make its way down into the ureters. All right, moving forward then. So moving forward here, we've gone through the different parts. So make sure you got those down. Let's talk then urine formation. Now when we talk urine formation is going to be a process that's going to get divided into a few different steps. Those steps include, number 1, glomerular filtration. Number 2, tubular reabsorption. And then number 3, we can see tubular secretion and tubular secretion. So let's talk first glomerular filtration. Now, when we talk glomerular filtration, I'd like you to know glomerular filtration is going to occur when our blood is going to enter the afferent arteriole and then into the glomerulus. Because of glomerular blood pressure, we'll see that blood pressure is going to be high in the glomerulus. Because of the high blood pressure inside the glomerulus, water and small molecules are going to move into the glomerular capsule. So we're going to have this process of filtration take place thanks to blood pressure. That blood pressure inside the glomerulus is going to force water and small molecules into the capsule. So that blood pressure forces filtration to take place. It's going to force filtration to take place. This is a filtration process because we'll see large molecules and formed elements are unable to pass through the capillary wall. So it proves that to you. So it's safe to say blood in the glomerulus has two components. Number one, the filterable components, which are going to include water, nitrogenous waste, nutrients, and salts. And then the second component are going to be the non-filterable components. The non-filterable components are going to include components such as our formed elements from our blood, our formed elements, or you can say our blood cells, and our platelets, and our plasma proteins. So those will be the non-filterable components. The nephrons we'll see are going to be responsible for filtering small molecules such as glucose and ions such as sodium. So here, let's check this all out. So again, here are all three processes. Glomerular filtration. Now the components that get filtered, you can see here now in greater detail. So all these components get filtered out into, we said, that glomerular space. From there, then they're going to be routed into the PCT. So the next process that's going to occur then is going to be tubular reabsorption, and then tubular secretion we're going to talk about as well. So here are our filterable blood components you need to make sure you know about, and then the non-filterable components. So these things should not be in our urine, right? We should not have any blood cells. We shouldn't have any red blood cells. We shouldn't have any platelets. We shouldn't have any white blood cells either. Nor should we have plasma proteins inside of our urine. Okay? Next in here, when we move through, here you can see how much water is filtered per day, how much is excreted, and how much gets reabsorbed. Same thing here we can see with sodium, glucose, and urea. So you can see nephrons are going to filter about 180 liters of water per day. Along with that, a considerable amount of small molecules and ions. The remaining processes are going to alter composition of the filtrate. They will alter the composition of the filtrate. So let's go into those remaining parts so we can check out the remaining processes. So next then we have what we call tubular reabsorption. Now when we talk tubular reabsorption, this process is going to deal with molecules and ions that are going to be passively and actively reabsorbed from the nephron, from the lumen of the nephron into the bloodstream, or you can say into the blood via the peritubular capillary network, thanks to the peritubular capillaries. We'll see about 65% of the total sodium that's going to be filtered by the glomerulus is going to be reabsorbed at the proximal convoluted tubules. Nutrients such as glucose and amino acids, they're also going to return to the blood thanks to the proximal convoluted tubule, thanks to the PCT. This reabsorption process is going to be a selective process, and only molecules that are recognized by carrier proteins are going to be reabsorbed. If they're not recognized, they won't be reabsorbed. Now, when we talk glucose, glucose you'll see is going to be an example of a molecule that is completely reabsorbed because there are going to be enough carrier proteins for it. Well, that's what we used to think. Now, this new Western diet we have, it's packed with glucose, right? Give me a number 4. Give me a number 7. Give me a number 8. By the way, supersize it, right? I mean, 20 cents more, might as well. Well, what is that supersized stuff? French fries, glucose. Soda, glucose. So now we've actually got too much glucose coming in. So with this too much glucose coming in, we don't have enough carriers there to make sure every single component of it is going to get reabsorbed. So what happens now is some molecules of glucose are going to be found in the urine. So every substance, I want you to know, has a maximum rate of transport. Glucose is no different. So after all the carriers are in use, any excess molecules, whatever we may be talking about, they're going to then appear in the urine. That's how we're diagnosing diabetes mellitus type 2. Now we've got even more smarter and more specific. We can go and we can check your hemoglobin A1C. And that tells us, so no matter what the patient says, your glucose for the last 90 days. When all of this glucose testing started, before they would actually taste urine. And there was a time also when they would just spill it and watch and wait and see if ants came to it to diagnose it. So here we've gotten smarter and we've seen and we understand now physiology in greater detail. So those receptors, they're said to be saturated. They're said to be saturated in diabetes mellitus. Again, any excess molecule then will appear in the urine. So if it appears in the urine, we say the receptors are saturated. They're all doing what they're supposed to be doing. We just have more of that molecule than we anticipated. In diabetes mellitus type 2, excess glucose appears in the urine because liver and muscles can't store it. They can't store it. The kidneys, they also fail to reabsorb all of it. So what happens is it appears inside the urine. It appears in the urine. So we can see that right inside of here. Now with all this glucose unable to be reabsorbed, this is going to cause increased osmolarity in our filtrate, causing water to be reabsorbed into the peritubular capillaries, which then is going to lead to frequent urination and thirst in the patient. When you talk about the telltale signs of diabetes mellitus, they include frequent urination and frequent thirst. Along with that, polyphagia, frequent hunger. So here we can see the reabsorbed components and the non-reabsorbed components. The reabsorbed components will include mostly water. The reabsorbed components will include most water, nutrients, and required salts. The non-reabsorbed filtrate components will include some water, most nitrogenous waste, and then excess salts. And then the third process is called tubular secretion. Now, when we talk tubular secretion, we'll see tubular secretion is going to be a second way by which substances are going to be removed from the blood into the peritubular network and added to that tubular fluid to be removed as urine. Hydrogen ions, potassium ions, creatinine, and drugs are going to be removed from the blood. And then in the end, you can see urine is going to contain two things: filtered substances that have not been reabsorbed and substances that have been actively secreted. So urine is going to contain two things. Filtration occurred. Filtration took out whatever it wanted to. Whatever didn't get removed went back in the blood. So now the filtrate in the tubules, whatever gets reabsorbed from it goes back into the blood. It's not going to be inside that filtrate. And then we said substances that have been actively secreted. So whatever gets secreted out that didn't get filtered in the beginning is going to also be making up urine. So again, it's filtered substances that have not been reabsorbed and substances that have been actively secreted. Another very fascinating system. Very fascinating. Thank you.